is reproducible research a holy grail? And of course, the dirty secret of science is that the vast majority of research is not reproducible. We still seem to manage. So what is reproducible research? And there is some confusion about what people mean by reproducibility. The language actually matters here. So do they mean it's repeatable? Do they mean it's replicable? Do they mean it's reproducible? Do they mean it's reusable? These are quite distinct concepts. So repeat means I can actually repeat it in the same setup, the experimental setup with the same authors, and it wasn't a fluke. Replicate means that I can run it under the same experimental setup, but by people who are not the authors. Reproduce means I may vary the experimental setup, so under different conditions. The data may be different, the experimental methodology may be adapted, but it's basically, can I reproduce the results that were there, but under a different setting? And then reuse will be, I want to use this method. I want to use this data in order to do something else, to build on as part of the collective knowledge of the community. And when we're saying reproducibility, we mean all of these things at different stages. And of course, there are different steps in the review process. Replication is a step in order to be able to test this, to certify the claims of the paper. This is to stop you saying something is true when it is not. So it's checking, is this a fraud or not, effectively, or was it a fluke or not? Whereas reproduce is a step to do comparison. It's a step to do, can I compare an approach with another approach? Can I use my data with this particular methodology, for example, or run this protocol in my laboratory with my cell lines and see what happens? So that's quite different. So replication, that's what we want when we want to accept papers into the publishing pipeline and be sure that they're honest, and it's almost impossible to do because it's so expensive. Where's the journal of reproduced results? There is no, who reproduces those results? PhD students and postdocs in the lab who are trying to use the protocol. And their contribution to a post-publication peer review will be jolly useful, because all that's hidden in notebooks at the moment, and it would be great if we could get it attached to the papers. Whereas reproduce, that's actually what people actually are doing, reproducing and reusing. So I think replication is a pipe dream, and reproduce is what we do. Open data is one of the key tools that we can use. So you need at least 4 things to be able to reproduce something. You need to have a very good description of the methodology, sufficient that you can really run the methodology. You need to have open access to the components. So that could be the data, be the software, but you need to make those available to you. And I work a lot in computational science. I am a computer scientist, so everything I do is computational. Then you need to be able to port the whole experiment. So in computer science or computational biology, where I largely work, this is porting the entire software or porting the entire data set so that you can effectively run it in your laboratory, which is like run it on your computer. Porting an entire lab is something else altogether, a wet lab. You can't do that, but you can do it computationally. You can do that. You can port the software. And then the 4th thing that nobody does properly, in my experience, is have a clear description of the degree of variation and the boundaries of tolerance. So to what degree does the setting have to vary before it can no longer be held valid? And in computational biology, this means to what degree has the software migrated so much, or the algorithms have migrated so much, or the data under which the experiment was previously, the analysis was previously done, varied so much and changed that you can't compare like with like anymore. And hardly anybody describes variation in their paper systematically. So those four things, and then you're good to go. And what Gigascience, which is a lot, by the way, what Gigascience is doing is creating the portable platform, which is the software environment in order to be able to access the data and run the protocols. Is providing a very strong description of the methodology. It's describing open data. It's providing open data and open software. So it's providing at least three of those components. And if you've got three, you can do something.